Chapter 19, we're going to look at several concepts that are associated with evolution: speciation, taxonomy, systematics, and phylogenetics. So first we'll kind of wrap up this evolution and then talk about speciation. First, kind of introducing what a species is, things like that. So mutations are the raw material for evolution. The DNA nucleotide changes can alter how proteins function or how they are expressed, creating new phenotypes. So as we had learned before, with natural selection, sexual selection, all of these different ways that evolution can arise, these new changes that are positive are created through mutation. So a new phenotype or a new genotype can form through mutation if it happens to be beneficial or associated with something that is beneficial. Then it can persist in the population more and more through its frequency. Evolution is what is creating life's diversity. As we learned with microevolution, these are small changes in the population that are accumulating over generations. So from one generation to the next generation to the next, this is microevolution. These are small changes in the gene frequencies, and they can swing back and forth. A particular gene can become in high frequency and then over a few generations become low, and a few more generations become high. It all depends on the environment. If the environment is changing, then these frequencies will change on a regular basis. Over long periods of time, these small changes can accumulate to become very large changes, and this is macroevolution. This is slower and results from large-scale changes. It can include, and many times does, include new species or many new species. As new species form, lots of others can become extinct. It could be an even ratio of new species to extinct species, or one can be higher than the other. It all depends on how many niches are out there to be filled in the environment, what kind of small parts of the environment can be exploited by these new species. So what is a species? A species is a distinct group of organisms, but over time, the definition of species has changed many of times. Early scientists defined species by the way they looked. If they looked similar to one another, then they would be lumped into the same species. Early scientists like Linnaeus and Darwin were looking at the morphological characteristics of these organisms and grouping them together into species. In the 1700s, Carlos Linnaeus was the one that created the naming scheme for species that we use today. We call this taxonomy, and this is the whole domain, kingdom, phylum, class, order, family, genus, species. And lots of this we still use today, but it is constantly changing. We're finding new relationships and different groupings that should be, and things like genus and species are changing pretty regularly for certain groups of organisms. So each species will have a name that is combining the broader genus and the more narrow term species together to create a scientific name. For humans, our scientific name is Homo sapiens. Homo is our genus, sapiens is our species, and together it is Homo sapiens. Now, you might have heard the term Homo sapiens sapiens, and that last sapiens is actually a subspecies. So we are a subspecies of Homo sapiens. I won't get too much into subspecies, but it's basically different groups of this one particular species. It's still considered the same species, but there is some morphological differences, usually over space. Their populations are defined by particular characteristics. To the left, or sorry, to the right, we see two cats, the leopard and the bobcat, and these organisms as well also have a species, a scientific name of a genus and species. The leopard is Panthera pardus. The bobcat is Lynx rufus. So we still use the Linnaeus taxonomy for naming of groups of organisms, but more modern definitions of species have come about, except for just the way they look and sharing appearances. Modern biologists define species by reproduction. If they are able to reproduce, then they must be the same species. So this is the biological species concept, defined by if species based on their potential to interbreed and produce fertile offspring. So not only able to produce offspring, but that offspring must be able to produce offspring as well. New species form when some individuals can no longer interbreed with the rest of the group. So if a group of the population is no longer able to interbreed with the larger group, then they would be considered a new species. We'll look and see how that can happen. So this is a great definition, but there are lots of organisms that are still very difficult when using that definition. Are they the same species? So lots of species cannot be defined by reproduction. So if we think asexually, so organisms that reproduce asexually, there is no can they reproduce with one another because they're creating their own offspring. And so how do we define species there? Fossils. So when we're looking at fossils and trying to figure out if these fossils are the same species as these fossils, well, there's no way that we can see if they interbreed with one another. So that falls apart at that situation. Some organisms are able to interbreed in nature. Some organisms are able to interbreed in the lab, but out in nature they never do. So if you take two individuals and you kind of force them to be together, they might be, let's say the liger, which is a combination of a tiger and a lion. In nature, tigers and lions, their distribution areas do not overlap. Tigers are in Asia, lions are in Africa. But if you put them together in an enclosure, sometimes they can interbreed and have a liger. But in nature, that wouldn't happen. Later, we'll find that liger is not fertile. It's a sterile offspring. Lots of even more modern ways of defining species is by using DNA analysis. So the similarities of DNA, how much similarities of DNA, how much differences exist. So researchers compare the nucleotide sequence of genes that organisms have in common. And so sometimes the amount of DNA that's actually being transcribed and translated into genes is a very small amount. And so sometimes looking at this similarity in the nucleotide sequences, they might be looking just at particular genes, just at particular portions of the genome that create proteins, instead of the entire genome. But different organisms, they look at different things. Different organisms have specific things going on with them. In bacteria and in archaebacteria, if DNA of the organisms is more than 97%, then they're considered the same species. So when it comes to bacteria and archaebacteria, these organisms are reproducing asexually. And so to define if they're the same species or not is based on the similarity of their DNA. But when we look at humans and chimpanzees, we share about 99% of the same DNA. When looking at the genome of particular parts of the genome, when looking at the entire genome, we're about 96% identical. But just the fact that we're 99% identical in the parts of our genome that create proteins, I mean, that's more than 97%, so we're obviously not the same species. So when looking at different organisms, this 97% is not the same number across the board. Ernest Mayr in the 1940s wrote a paper kind of talking about defining speciation. He wasn't the first person to coin the term speciation, but he was the person that kind of lifted it off the ground and really made it a thing. So speciation is the formation of new species. And so we'll be looking at how speciation can take place, and it is when a population can no longer successfully interbreed. So really, it would be that there is some sort of a reproductive barrier, some sort of a splitting of that population into two populations. It could even be multiple. It could even be more than two, but usually two is the way of thinking of this. So the population is split into two. There's some sort of barrier between them so that they're not interbreeding, and there are two distinct gene pools. Well, over time, mutations will take place in each of those gene pools. The mutations are happening randomly, and so they will not be, the same mutations will not be taking place in both gene pools, and because there's no interbreeding between the gene pools, that mutation will stay just in that one particular gene pool. Over time, more and more mutations take place. It's just natural, and with enough time, the genome of each of those populations is going to be so different that if you put them back together, they will not be able to interbreed. This is the concept here, and we'll look and see how some of these processes take place. So this requires reproductive isolation. Reproductive isolation is some sort of a barrier that is going to stop these organisms from interbreeding. And so it could be spatial, time, actual mechanical, their physical parts, their gametes aren't coming together, things like that. And we'll look at these. So reproductive barriers can cause speciation. Now those, if enough time does not go by and those two populations are put back together, they could create viable offspring. Every species, every situation requires a particular amount of time that is unknown to us. But with that time passing, that amount of change in their genomes taking place is what will cause this new species. So if the potential to interbreed defines species, then reproductive isolation results in new species. Now, just because two organisms are physically separated from one another, let's say a river cuts through a population of worms, just because a population of worms is on one side of the river and one population is on the other side of the river, doesn't mean that they're all of a sudden now two species because they're not interbreeding. It's the ability to interbreed. So if I were to scoop up some worms on one riverbank, and take it to the other riverbank, well, they would interbreed easily. It's the reproductive isolation and time that's going to create this new species. So reproductive isolations can be either prezygotic, which is before the zygote or before the embryo, or these reproductive isolations can be post-zygotic, after the zygote or embryo has been created. And usually that has to do, this hybrid of the two groups, this offspring that has been produced will either be sterile or will have reduced fitness. In the next couple generations will not be able to produce offspring. So this page has a lot going on, but this is a list of the prezygotic reproductive barriers that prevent fertilization. So there are a list of 5 here. The first three, if they exist, they're not necessarily going to produce a new species. It's going to take more time. As we get further down in the list, then it's a bigger chance that a new species is existing here. And so prezygotic is before the zygote is formed. Behavioral, temporal, these things like this. So the first one that we'll look at here is the barrier is habitat isolation. So these organisms, this population, is splitting up their habitat. They're living in different environments. And so that could be small or large. Let's say you're in a mountainous area, and there's mountains and valleys. Well, part of the population starts going up the mountain to live, whereas the other part of the population is living down in the valley. And let's say that the mountain organism, the mountain part of the population, doesn't come back down for anything. It stays up there. Well, they're physically isolated. Their habitat that they live in is isolated. If they're not coming into contact with one another, then they're not going to be interbreeding. And so this is a physical isolation, a habitat isolation. Another example here is ladybugs. So ladybugs will basically, they can, different bugs, different organisms can live their whole life on a particular plant. And in this case, we've got ladybug population all living on this particular species of plant. This species of plant stretches across this area. Well, part of the population starts going over to another plant. So instead of the first population was dealing with strawberry plants, well, now this new population is jumping over to blueberry plants. Well, if that's happening, the population that lives on the strawberry plants will never interact with the ones that are on the blueberry plants. And so they have physically isolated one another, habitat isolation. The next one is temporal isolation. And so this is time. This could be time of the day. This could be season that they're active in. So if the population is active during the day, but then part of the population starts to become active at night, and the population splits to a day and night, yes, they're physically there with one another, but they're not active to mate at the same times of the day. Or a certain population, the population is coming out in the spring, and then they're not active anymore. But for whatever reason, part of the population becomes more active in the summer or even fall. They're again, they might be like hibernating during the same time, but they're not out and active during the same times. When we think of cicadas, we don't have a lot of cicada activity down here in Hampton Roads, but it does happen. And there's the 7-year cicada, the 13-year cicada, the 17-year cicada, and so these organisms come out in prime years, and they're different species. And so if you've got a species coming out every 7 years and one coming out every 13 years, well, it's going to take a really long time for them to actually come out at the same year. And so and they're related to one another. So this temporal isolation, behavioral isolation. Um, there's some sort of behavior that is stopping the population from mating with one another. And so one of the big ones here is the different courtship activities. If we think of frogs having a mating call that is going to attract their females of their population, well, let's say there's a male that all of a sudden starts creating a new call. And there's one particular female that likes that new call, and so they mate. And then part of that population, the offspring that they create, then are all doing this new call. The females are all interested in that call. And so over time, you could have these two populations that are living in together with one another, but the females of one population are only mating with the male croaks of that population, and the vice versa with the other population. So they will not be interbreeding. Now these are perfect examples. Perfect does not ever happen in nature, but they're still these are perfect examples. Um, so these types of things can lead to uh, populations not being able to interbreed with one another over time. When it comes to mechanical isolation, this is when the actual organs, the mating organs, do not fit with one another. So when we think of a penis and a vagina coming together like a key and a lock, so they perfectly come together and the key can open the lock. Well, as we all know, there's lots of keys out there. Not all keys will open different locks. And so if you put two species together, sometimes their physical mating organs cannot fit into one another, and they're unable to interbreed because they physically can't get the sperm to the egg. And it's also pollinators. So when we think of plants being pollinated by bees, that's how they are interbreeding. That's how the plants are interbreeding with one another. So if for whatever reason the pollinator, the bee, is unable to get to the pollen of that new population to fertilize the flower of the same population. Um, and then there is gametic isolation. So again, this is prezygotic, so before the zygote. The zygote is when the sperm and egg have come together and formed a fertilized egg, a zygote. So this is before that. So gamete isolation. The gametes cannot come together. So the sperm gets to the egg, tries to fertilize the egg, but cannot. There's no fertilizing happening. And when it comes to mechanical and gametic isolation, to me, this is pretty much they are a new species. If they are not able to interbreed because of these two reasons, then they're pretty much, they're not able to interbreed. And then these first three, yes, they're not coming together to interbreed, or they're not in the same areas to interbreed type of thing. But are they yet a new species, and that depends. That depends on the situation. Post-zygotic reproductive barriers, they're going to prevent the development of fertile offspring after the zygote, has formed. So the first one is hybrid inviability. So basically, a hybrid has been formed, offspring has been formed between these two populations, but that offspring fails to reach maturity. It ends up dying and not living to the time of maturity when it can reproduce. The next is hybrid infertility. So this is the offspring, the hybrid is created, but it is sterile. The hybrid offspring is unable to reproduce. And this is the case when I talked about the lion and the tiger coming together to produce a liger. The ligers are infertile, they're not able to have offspring. Another example of this in the world is mules. Mules are an offspring, a hybrid of the donkey and the horse. The donkey and the horse are two different species. If they have offspring, you'll have a mule. Mules are infertile. They're not able to have their own offspring. And so this is kind of the same situation. Then we have this hybrid breakdown. So the first generation of hybrids is formed. They're able to create offspring, but then that second generation will not be able to create offspring. It will die off. And then the example, they're talking about mosquitoes. That second generation will have this abnormal genitalia. The genitalia just won't be able to line up with the other species genitalia to create, to fertilize the egg, to put the sperm into the female. So these are the post-zygotic reproductive barriers. And basically, to me, these are, this is the new species. If this is taking place, if these are taking place, then you have kind of created this new species. This will be the reason that they're not able to interbreed. Speciation patterns, or I should say, there are three types of spatial patterns that define speciation. And so these are kind of work together with these reproductive barriers. This is how reproductive barriers can arise. And it will all depend on these spatial barriers. And so we'll talk a little bit more about each of them. We have allopatric speciation, parapatric speciation, sympatric speciation. So with allopatric speciation, populations have become geographically separated. They're physically separated from one another. Some sort of barrier, like physical barrier, is separating the two populations into groups that cannot interbreed. And so it takes a long time for mountains to form. But if we're talking about macroevolution, we can think of a mountain range has formed and split two populations into, split one population into two. And now, this would be an organism, not birds. This would be an organism that physically cannot or will not go over this mountain range. A river forming could be this physical barrier. If the as Pangea broke up and these continents are drifting apart from one another, that splitting of the continent can be a physical barrier. And so there will be no gene transfer between these two populations, each proceeding down its own evolutionary line. And so we can see how, since they're not interbreeding with one another, then you've got all these mutations taking place, and if they ever do come back together, if enough time has passed, they will not be able to interbreed. So allopatric speciation can create these reproductive barriers with enough time. Eventually, genetic differences between the populations give rise to reproductive isolation, either prezygotic or postzygotic. One example, we're talking about the tortoises on the islands of the Galapagos, and as storms can come through and kind of sweep some of these turtles away, and they can show up on a new island. You get a male and a female, then they can mate, and so once you have part of a population on one of these islands, it is physically separated from the other population, and with enough time, you can create new species. Now, this is probably not the greatest example because it's saying that these are subspecies of one another. On different islands, they're subspecies. So they haven't quite reached that speciation yet, but they are on their way. With enough time and not interbreeding with one another, they can create new species for each island. Parapatric speciation will occur when the population is still touching, is still neighboring, but the majority of the populations are not touching. So in parapatric speciation, part of the population has entered a new habitat, bordering the range of the parent population. And so, let's say there's a valley that has opened up in this mountainous area, and part of the population has moved into the valley, and they're living in the valley, but the rest of the population is outside the valley. Well, right there at the border of the valley, they're able to come together and interbreed. But just at that area where those two groups are coming together, the rest of the area, they're interbreeding with each other. The valley is interbreeding with the valley. Outside the valley is interbreeding with outside the valley. With enough time, you can get genetic differences on those extreme ends. You can get enough genetic differences that you can start to get speciation. Now, this is more of a reality. This happens much more than probably some of these other ways. Mating can occur between populations on the border, but most individuals mate in their own population beyond the border. So I have an example of mosquitoes for this one. In the north, we've got Virginia, Maryland, Pennsylvania, New York, up into New England. We have one species of mosquito, a Culex pipiens. We also have a population, Culex cuncifasciatus, that is living in Virginia, North Carolina, South Carolina, down into Florida. Well, as you can imagine, Virginia is that hybrid zone. Virginia and North Carolina are that hybrid zone of the two. And these species are our main West Nile virus vectors up and down the coast of the East Coast. And so they're able to mate and interbreed in the Virginia, North Carolina area, but that's where they border. When you go higher up or further down, they're their own particular species. But in our area, they're a hybrid. And the pipiens are they're really good vectors for West Nile up in the north. Cuncifasciatus is a really good vector down in the south. But when you look at the hybrids in our area, for whatever reason, they're not able to be as good of vectors for West Nile virus. So we're kind of in this nice area that West Nile does not persist as much as it does further north of us and further south of us because of this hybridization, because of this genetic difference. It's not either particular species, it's a mixture of the two. But they are able to interbreed. Those offsprings are able to be viable. So new species to the north and to the south, but they're still able to hybrid. So that kind of goes against the biological species concept that I was telling you earlier. Biological species concept works great theoretically, and scientists use it. But in reality, there are still lots of areas and groups that can hybridize on these borders when they come together. It's just, it is what it is. Um, yes, parapatric speciation can create these reproductive barriers. Genetic differences accumulate between the populations, which is making the interbreeding less likely. So as the populations, the genetic changing is happening on the extreme ends of the populations more, and the more that those changes are taking place, those changes kind of go through the population down to where the border is, and eventually at that border there might not be hybridizing happening anymore, or that hybridizing could happen for a long time. It all depends on the situation. I should say, all depends on the species. Parapatric speciation. Looking at an example here, the little green bulls. And this is a bird living in Africa. So two populations. Well, it started as one and then split into two populations where one of them was living in habitats like grasslands. The other population was living in habitats like forests. And so both of those areas are isolated from one another, except for right on the border where forest turns into grasslands and you have this kind of mixture. Well, this is where the two populations come together and can interbreed. But in the extreme ends of the population or the extreme environments, they're not coming together to interbreed. So yes, you could have this genome, you could have these genes kind of coming in, but they're not going to fill in. The forest could be huge, this grassland could be huge. Sympatric speciation is the last of the three, and this occurs in a shared habitat. So sympatric speciation, populations diverge genetically while living together. Although the habitat may appear uniform, often it consists of microenvironments that select for different phenotypes. So how sympatric speciation can take place? It could be those frog calls that we talked about when it comes to behavior. They are still intermingling with one another. They're still in the same habitat, but they are separating by their frog claw calls. Now, even the ladybugs that were separating their area by the blueberries and the strawberries, from our perspective, blueberries and strawberries could be in the same area, and so it seems to us like this is sympatric speciation, but when you zoom in and you get close, they're still physically separated from one another. Is that really sympatric speciation? Or is that allopatric speciation, where they're physically separated by the plant that they're on? Some people feel that sympatric speciation doesn't exist. There's always some sort of physical separation that is taking place if you look close enough. But we are learning that sympatric speciation exists because of situations like the frog calls and behavior situations. But as you can see in this picture, it's showing that they're all living together, even though they're kind of grouped in little groups. They are able to speciate sympatrically. One example here is the Kitchelid fish lives in Africa. This is some lakes in Africa, and both populations live in these lakes. One species of Kitchelid has diversified into two species in this African. Each fish specializes in a unique microhabitat. One of the species lives in the deep water of the lake, and the other species lives in the shallow coastline of the lake. And so they have specialized into these different microenvironments, and they're all living in the same lake, but there is still some physical separation, right? But this is considered allopatric or sympatric speciation because they're living in the same environment. They're all coming into contact to some degree, and select forces in the two different microhabitats has led to a reproductive isolation and then speciation. There are different paces of speciation. We looked at this earlier in lecture 17, gradualism and catastrophism. In this case, catastrophism is talked about as punctuated equilibrium. So the fossil record supports two hypotheses about the pace of evolution. Gradualism is the small incremental changes over many generations. Evolution and speciation are happening gradually. New species are forming all the time at a gradual basis in the history of Earth. Punctuated equilibrium, long periods of little change, and then those are interrupted by bouts of rapid change. And so really, both of these are happening. Gradualism is taking place in between these interrupted bouts of rapid change. So gradualism is happening. New species are kind of being formed. Again, how fast that's taking place depends on what organisms we're talking about. Whereas punctuated equilibrium, these interrupted bouts that are taking place are happening after a catastrophic event. Some sort of a major extinction has taken place, opening new environments, and or a volcano erupted, or different things, depending on what groups of organisms we're talking about. Bursts of speciation can occur during adaptive radiation, and so this is related to this punctuated equilibrium. A population inhabiting a patchy or heterogeneous environment gives rise to multiple specialized forms in a relatively short time. So if a species finds itself in this new environment, let's say it has never been on this new island, and it finds all of a sudden this island is opened up to this new species. Well, this new species comes in, they're going to be finding new prey, they're going to be finding new habitat to live in, and it's possible that it can give rise to specialized forms, which are going to be isolating themselves by habitat, by maybe different times of the day, depending on the prey, and those reproductive isolations can give rise to different species with enough time. And then the picture is showing down at the bottom the first flowering plants, for example, diversified quickly as flowers provided new options for reproduction. So as soon as this adaptive quality arose in flowering plants, all of a sudden they were opened up to these new opportunities, and they were able to reproduce. They were able to diversify quickly to kind of come into these niches and diversify. Punctuated equilibrium, fits very well with allopatric speciation. We have an example here. This lizard has come onto a new island in the Caribbean, and uh, it was able to diversify because of all of these different habitats that exist on this island. You've got populations, you've got a species living in the grasses, a species living on the trunks of the trees, up in the canopy of the trees, different times of the day, feeding on different species, feeding on different prey that is found throughout these areas, adapting physically to eating and living in those environments. Extinction marks the end of a species' existence. A species is extinct when all the members of that population have died. Most extinctions occur as part of a background extinction rate. This is basically, throughout time, new species are formed, new species die. It's a natural process. This background extinction rate is a pace which species go extinct due to gradual changes in the environment. The environment is always changing. We talk about major changes being human caused and negative, and this is true that humans are creating rapid changes to the environment quickly, but throughout the history of Earth, the environment is always changing, and some animals, some organisms are able to adapt fast enough not to die, and then some are not able to adapt fast enough and they will go extinct. Uh, there are mass extinctions throughout the history of Earth. And with those mass extinctions, we can have this punctuated equilibrium where lots of speciation will take place afterwards. So mass extinctions, major environmental changes can cause these mass extinctions. An asteroid hits the Earth, throws up debris into the atmosphere, which will change the temperature of the Earth dramatically, allowing not enough light to pass through. Photosynthesis takes halt, and things like this can dramatically change the Earth to the point that there are mass extinctions where 95 to 99% of certain organisms have died because of this mass extinction. The surviving organisms are able to exploit these new resources. So as the organisms die, they're opening up these new niches, and those new niches can be exploited by what is already existing there. So it's just like these organisms that are still living, instead of being introduced to this new island where they can radiate out in diversity, they're just being thrown in new opportunities from the land that's already in front of them. They can exploit these new resources in the changed environment, and they can just diversify after the others have. Many species are lost quickly in mass extinctions. The fossil record shows evidence of 5 mass extinctions over the last 600 million years. 600 million years is basically when the Precambrian meets the Cambrian, and that's kind of when most of our multicellular life kind of formed. It was around 600, 500 million years ago. And since then, there's evidence in the fossil record to show 5 mass extinctions, and these were due to major environmental changes. The 6th mass extinction is happening right now. Current extinction rates are accelerating due to human activity, especially in vulnerable areas like islands. So when we think of human activity affecting extinction rates, we think of what we're doing right now. We think of cutting down habitat, for deforestation, fragmenting habitats, so building roads and things in between pathways from one habitat to another. pollution that is devastating the earth, acid rain, um, different the carbon emissions, methane emissions, all these different things. We're introducing non-native species to different areas around the world. We've introduced rats to everywhere in the world. Cats, which is a predator to many small animals all over the world. These cats exist. The lionfish, which is out competing native fish. Mosquitoes that are introducing new viruses to different populations of birds, mammals, humans. But humans have been doing this for a long time, not just modern humans. When we think of when Americans or American Indian ancestors came over to North and South America around 40,000 years ago. They actually came a couple different times within those 40,000 years. During ice ages, the ice would freeze over, or the oceans, the Strait of Gibraltar would freeze over, not the Strait of Gibraltar, the Bering Strait between Russia and Alaska, would freeze over and allow humans and other animals to cross over from Europe to United States, or sorry, Europe into North America, or vice versa, North America into Europe. And so humans came over and they found large mammals. North and South America looked like Africa, with large woolly mammoths that are similar to elephants, giant sloths that stand 15 to 25 feet tall, huge bears, huge wolves, all these large animals. And they were able to reap those rewards and hunt those animals, get that food, get that that fur for using for warmth and things like this. Well, over time, it killed a lot of these large mammals and caused lots of mass extinctions or lots of extinctions to these large mammals. They cultivated and changed the landscape of the plants, altering and changing and killing certain animals and plants along the way as well. So not only are modern humans the cause of this mass extinction, this sixth mass extinction, but humans building up to what we are today have been a cause of that now. What we're doing today is probably a lot worse. But I mean, that's, it's happening all over. Other reasonings for mass extinctions in the past. Humans are just the cause of this sixth, but what about the other five? Catastrophic events explain these mass extinctions. Some of them are caused by this impact theory, which suggests meteorites or comets hit the Earth and cause these large extinctions. So a meteorite hits the Earth, throws up not only the organisms that live close to this impact are going to die pretty soon. Anything that's far away, the dust and soil goes into the atmosphere and will stay there for years, blocking the sun from coming through and stopping photosynthesis, affecting the world greatly. And it's the entire world when a big meteorite hits. It's not just what's close by. The dinosaurs went extinct because of such an event like this in the ending the Cretaceous period. So these mass extinctions kind of are the beginning and ending of certain eons, eras, periods in time, in a geologic time, because they are causing the fossils to be extremely different. So right before the event, the fossils were of these particular animals, let's say dinosaurs. After this event, the dinosaurs have died, and now you've got this diversity of mammals. And so it's not like this event happens within, moments in reality. It happens probably over lots of years. But in the geologic time frame, in the fossils, it looks like it happens in just one step in the soil. These impacts change the environment, like I was saying. Debris suspends in the atmosphere and drastically changes the environment, leading to the extinction of many species, sometimes 95% of terrestrial and even aquatic animals and plants. Plate tectonics can help explain mass extinctions. So, Earth's shifting land can have major effects on water and land. And so the temperature of the Earth is going to be affected if you have one giant landmass like Pangea, or if that landmass is spread out. Just a few degrees can dramatically change what's going on Earth. And if organisms cannot respond fast enough, then they can go extinct. You also have lots of volcano activity, earthquake activity that are affecting organisms' lives. Volcanoes, just like an impact of a meteorite, can throw up ash into the atmosphere. And if you have enough volcanic activity, you can dramatically change the climate for a long period of time. Moving on to the next topic, which is taxonomy. So we had mentioned Carlos Linnaeus earlier. He devised our current classification system, and this is a way of organizing life's diversity. He did not know anything about evolution or natural selection. He was basically looking at the morphological characteristics of these organisms and classifying them, putting them in groups compared to their similarities. Because Linnaeus did not know about evolution, he did not know that organisms have common ancestors and descended from one another. So, in the early taxonomy, he did not take these into account. More modern taxonomy tries to take these factors into account when grouping new or changing these groups. But some of the fundamentals of some of the larger groups do not take into account the common descent here. So some will, and we'll learn about some classification systems that are based on common descent. But this taxonomy way of doing things, Carlos Linnaeus's taxonomy, is not fully based on common descent. It does seem that way in a lot of groups, but that is not how it was started. Modern biologists try to organize all species in a classification system that reflects evolutionary history. So modern times we try to do this because we understand. But we are still using an older system. This taxonomy is the science of describing, naming, and classifying animals, species, all species. And so the taxonomic hierarchy organizes species into progressively larger groups. If we started at the bottom, we have a single species, aloe vera, a species of plant that we get aloe from, that you put on your burns and different wounds and things like that, and helps. That juice is found in the actual stalks and leaves of this plant. And so you have one species, aloe vera, and that species is lumped into a genus of 500 species. And the genus is aloe, and that's where you get that aloe vera. And then you take that genus and you put it into a family with other genera, and you and you get the 785 species lumped into this particular family, and so on. This family is then grouped into an order with other families. This order is then lumped into a class with other orders, and each of these get more and more species. These are kind of the negative is saying that there is roughly that many species. When looking at an organism that we're all a little more familiar with, the domesticated dog, they also fall into this taxonomic hierarchy like all organisms on Earth. They are artificially selected, yes, but they still are an organism found on Earth. And so they are found in the domain Eukarya, the cells with organelles. In that domain, they are in the kingdom Animalia. They're not plants or fungi or protista. They are Animalia. In Animalia, the phylum Chordata, the class Mammalia. And so up into this moment, we also fall into this. We are also in the class Mammalia. Well, then you get into the order Carnivora. We are not in that order. We are primates. And so carnivora includes bears and cats and dogs and wolves and things like this. Then the family Canidae. And so Canidae is a lot smaller of groups. There are not as many, but there are still lots of canines, wild canines around the world. Then the genus Canis, and then the species Lupus. It used to be thought that domesticated dogs were a different species than wolves. And so domesticated dogs used to be Canis familiaris. But that has changed over the last couple decades, and they are now considered Canis lupus, which is the same as wolves, but they are a subspecies. They are Canis lupus familiaris. And so that is the hierarchy here of the domesticated dog. Taxonomy organizes species into groups. Modern taxonomy is based on the work of Linnaeus. We are building off of Linnaeus's group of work. The more features that two organisms share in common, have in common, the more taxonomic levels they will share. And so, as you can imagine, the chimp and the orangutan are going to share lots of taxonomic levels because they have lots of similar features. Whereas the squid and the fly, very different from one another, they are going to be very different in their taxonomic levels. They're both eukaryotes, they're both Animalia. But then once you get from that kingdom, once you get into the phylum, they will be different. But remember, taxonomic groups do not reflect evolutionary relationships. A lot of the times these features that they're based upon will also be evolved together. And so when you look at these taxonomic groups, most of them do reflect evolutionary relationships. But taxonomy itself is not looking at evolutionary relationships. It's only looking at the morphological characteristics. But some biological classification systems do look at common descent. They are based on common descent. Phylogenetics is a study of evolutionary relationships among species. So in a phylogenetic tree that we see on the right, we are looking at evolutionary relationships based on the descent of common ancestors. So we have here a list of 10 species. They are on the terminus branches of this phylogenetic tree. And we see that when the branch happens, so this is, it doesn't say this, but on your x-axis, the bottom, this is time. And it's not a precise amount of time because not all of these branches are happening at the same amount of time, but it's just showing time in general. And so these two, species 1 and species 2, have a common ancestor, and that's where this branch is right here. This branch right here is showing a common ancestor between species 1, 2, and 3. This one is labeled. This one shows a common ancestor of 1 through 4 because all of them are attached. Now this one here, this is only a common ancestor for 6 and 7, but if you go back to here, it's a common ancestor to 1 through 7 because all species can be linked back to this one terminus, or I'm sorry, this one junction. And then here is the common ancestor to all species 1 through 10 that is listed. Cladistics is a portion, it's a subset of phylogenetics. So cladistics is looking at derived traits, shared derived traits. Cladistics distinguishes between traits that are ancestral or inherited and traits that are derived. They are new. Derived is something that shows up new, and they are not inherited. And so when looking at a cladistics tree, we see that this is for mammals. So mammals, there are 3 groups, monotremes, marsupials, and placentals. We should know what marsupials are, kangaroos, koalas, American opossum. Placental is most of the other mammals that we know about with a placenta. And monotremes is a small group. It includes the platypus, which I believe there's only one species of platypus, and the echidna. Echidna is kind of looks like a porcupine, but it is a monotreme, and I think that there are 4 species of echidnas. Most of these are down in, I think, Australia and some islands north of Australia. I believe that's where they all live. And so these are the 3 groups of mammals. And so when we're doing a cladistics tree, we are looking at the traits of mammary glands and hair. Well, all mammals share this. And so this ancestor here had it, and so every ancestor afterwards has it. Well, then the derived trait of giving birth to live young, and that is seen here. It is a derived trait that is shared by marsupials and placentals, but not the platypus. And so if anybody knows, or I should say not the monotremes, anybody knows anything about a platypus and echidnas, you know that they lay eggs. They are mammals that lay eggs. Now, they're a different type of egg than bird eggs and lizard eggs or reptile eggs. They're like a leathery kind of an egg, but eggs nonetheless. They are not giving birth to live young. And then, so this is a derived trait. And then this next derived trait is the placenta. So giving nourishment to the young inside the uterus. So this derived trait happens, and yes, humans are placental, but there are lots of others, dogs, cats, bears, elephants, all these other groups, whereas marsupials do not have this derived trait. The young is born, and then it crawls into the pouch and attaches to the teat, and it needs that nourishment. It gets that nourishment from the teat. Whereas the placental animals, mammals, are able to do this inside the womb. A cladogram, which is basically a cladistics tree. So a cladogram is a type of phylogenetic tree. A cladogram is a subset of phylogenetic trees. There are lots of different phylogenetic trees that have been created over time. What we're seeing here is what we use mostly now. This is a diagram. This diagram is a tool used to develop a hypothesis about the relationships between groups of organisms. So instead of seeing on the right-hand side just a list of species 1, 2, 3, 4, 5, we're actually seeing a group of organisms. So instead of an actual species, it's a group of them. I mean, mammals is a large group. Turtles, large group. All of these are huge groups, but they can be that way instead of a species. They can be written as a group of organisms. So on a cladogram, a basic unit is called a clade. A clade is a group of organisms consisting of the common ancestor and all of its descendants. So if we look at birds and non-avian dinosaurs in conjunction with this ancestor, this is a clade. So if you did not know that birds evolved from dinosaurs, this is your introduction. Birds and dinosaurs together form a clade. They are all the descendants and the common ancestor. So this is a clade. If you then include crocodiles and this ancestor, and birds, dinosaurs, crocodiles, all are a clade as well, because they form a group that has the common ancestor and all of the descendants. If you look at this entire cladogram, it is a clade as well. If you look at birds, dinosaurs, crocodiles, lizards and snakes, turtles, mammals, amphibians, and this ancestor here, all of them create a single clade because you have all the descendants and the common ancestor together. Common ancestors indicate evolutionary relatedness. According to this cladogram, dinosaurs, birds, crocodiles make up one clade. Within that, birds and dinosaurs make up a smaller clade. So I had talked about these earlier, but it puts it in writing. Cladograms identify the last common ancestor. So if you wanted to know turtles, snakes, and lizards, where their common ancestor was, you could trace back to the lineage from right to left until they intersect and find the common ancestor. So that's what these red dots are doing. You're following back to see where that ancestor is. Now, if you were to say that lizards and snakes and turtles are a clade, you would be incorrect, because you would have to include crocodiles, birds, and dinosaurs in order to have a true clade. Now, the way that last cladogram was, it said reading from right to left. And that's just because that's the way that cladogram was drawn. There are lots of ways to draw cladograms, and they can all be the same cladogram. They can all have the same information. So these cladograms all below are showing the same evolutionary relationships. They're just slightly drawing it differently. So we're looking here now at this, the mammals, the monotremes, marsupials, placentals. All of these are showing that. And you can see here, it's drawn kind of the way that we saw before. But this right here, what if you flipped it? The placentals and marsupials this way. It's the same thing. It's telling you the exact same information. Well, what if we flipped it all together and we had marsupials at the top? As long as these branches are connecting in the same way, it's the same cladogram. So you see how we just flipped this whole thing, and now marsupials, placentals, monotremes. So it's all of three of these at the top are the exact same thing. They're just putting them in different orders. There's no true hierarchy here of this one doesn't have to be at the top or the bottom type of thing. Like it can be moved around. Instead of drawing them like this, you can draw them with intersections like this. It's the same thing. Instead of a horizontal, you could have a vertical and draw it like this. Or you could draw a vertical with this shape limbs, and it would look like this. All of these are the same. To build a cladogram, you tally up shared derived features. So you'll be groups sharing the greatest number of derived features are hypothesized to be the most related, the most closely related. And so you'll take these features and you'll put them out into this graph here to kind of draw your cladogram. As you start to accumulate more and more features, this is working out perfectly. Sometimes they don't work out as perfect. There are computer programs that can do these cladograms for you, and they hypothesize or they create the most logical cladogram. And so what's happening here is we've got the features, the derived features here on the left, and then the different species, the different fossils, the different groups on the bottom. And when these features show up, you start checking them off, and then you can create your cladogram. And so with this one feature, all of the species it is found in. So that means that this characteristic, this ancestral trait, is found with all the different species. And so it would be put here because this ancestor here must have had it because all of the descendants have it. When you look at the next feature, this wishbone, breastbone, and the loss of the fourth and fifth fingers, this guy does not have that, whereas the rest of these do. So that means you know that this ancestor here must have had it because all of its descendants have it. And so on until you get to this last feature, feathers, arms as long as or longer than legs. And the only species that had this here was, modern birds is not a species, but a group of organisms in this species. And these are all dinosaurs here, except for the modern birds. And so that you know that this common ancestor between these dinosaurs and these birds had this feature. And so this is how you this is how you create a cladogram. Back in the day, humans used to do this by hand. Now we're able to use computers to create much more complex cladograms. Cladograms do not have to be totally based on morphological characteristics. You can put in DNA sequences and amino acid sequences of proteins, and it can create a cladogram as well. So this cladogram is comparing variations in DNA sequences. DNA can be obtained from living and extinct species, vastly increasing the amount of information that can be used. And so what's happening here is it's looking at the code of the DNA. And so this code can be thought of as characteristics, and then as a derived characteristic shows up, this is a point mutation, point mutation, point mutation. As these mutations start to add up, as they start to happen, the rest of these individuals share that. So you see that at position 6, A turned into a T. Well, all of these species, that happened, that's been passed on to the next generations, but it's not found here. And so a computer program can figure out and create this cladogram based on the sequences of DNA and how, if a mutation took place, every species after that descendant will share that mutation. And you can imagine that this can get very complex when looking at gigantic lines of code of nucleotides. Many traditional groups of organisms are not clades. And this is why taxonomy, using the morphological characteristics for taxonomy, is not giving us perfect clades, perfect groups that showing common descent. And I'll show you why. A clade for here, looking at the same clade, or looking at the same cladogram, amphibians, mammals, turtles, lizards, crocodiles, dinosaurs, and birds. A clade is a monophyletic group, one phyletic group. And so, the birds and dinosaurs, that is one clade, that is also a monophyletic group. This large birds, dinosaurs, crocodiles, lizards, snakes, and turtles, this is also a monophyletic group. It has all of the descendants and the common ancestor included. The term reptile is a paraphyletic group. Reptiles are crocodiles, lizards, snakes, turtles, but this is not a clade because we're not including birds. Now, even some people say that dinosaurs are reptiles, but no one says that birds are reptiles. Those are two big groups in taxonomy that are not put together, and so that's a big no-no there. And so a paraphyletic group includes a common ancestor but excludes some of the descendants of an ancestor. And so when we look here at what is filled in this gray, these are reptiles, but you have to include the birds if you want it to be a monophyletic group. Ectotherm, these are warm-blooded animals, opposed to ectotherm, which are cold-blooded animals. But if you look at endothermic animals, if you wanted to classify that as a group, it would be polyphyletic group. A polyphyletic group excludes the most recent common ancestor of its members. Birds and mammals are endotherms. They're warm-blooded, but their common ancestor was not endothermic. And so mammals are warm-blooded. Birds are warm-blooded. It's even thought that lots of dinosaurs were warm-blooded, but not all of them. So it's kind of being excluded here. So if you were to group birds and mammals together because they're endotherms, this would be polyphyletic because you're excluding all of these other descendants, plus you're excluding this common ancestor between birds and mammals. Now we know that this common ancestor was not endothermic because all of these other descendants were not. So that means when we think of characteristics that they all share, they're homologous features. Endothermy is an analogous feature. It was created at different times, probably from similar environmental pressures, and it derived differently in these different groups. So this, using endothermic as a feature for a group of organisms, mammals and birds, makes it polyphyletic. It is excluding the ancestor and all of these other descendants.